Good morning everyone. Today in this video I'll be talking about this eminent scientist who had contributed so much in the field of medical microbiology, immunology, hematology. Okay, so point wise I'll discuss about his contributions. So first I'll start with invention of magic bullet theory or magic bullet concept. Now what is magic bullet concept? Bullet means something which will specifically target to someone and destroy that particular part and not causing any kind of a harm to any other part. Okay, that is the bullet, that is the function of the bullet. Now why magic term, why it is used with this bullet? This specific terminology is applied to chemical compounds in case of medical microbiology. This concept is basically defines any kind of a medical compound that specifically targets pathogen, means those that cause disease in our body. Okay, pathogen, that will target the pathogen and destroy them. Destroy what? The pathogen only, without causing any kind of a harm to host. So this specific chemical compound that specifically targets pathogen only, that is why it is known as bullet. And as it targets and kills the pathogen only without causing any kind of a harm to their host, that is why it is known as magic. There is a magic, there is a tricks. So this is the magic bullet concept. Now the second thing, that is basically side chain theory in immunology. Side chain theory, I'm not writing here the full term. Side chain theory of immunology. This is basically the body parts of human body that have various cells. He told that, or his concept was, cells have many side chains. Actually these side chains nowadays are known as receptors. So cell receptors, they are present on the top of the cell surface. So that is the side chain according to Paul Ehrlich. Then cells have the side chains that will interact with the toxins. If any kind of a toxins or foreign particles enter into our body, that toxin that will react with this receptors or the side chains, and that in turn give rise to more side chains. Okay, now these side chains, they will act as antibodies. Now what is the function of antibody? They obviously kill that toxin or neutralize that toxin and produce the immunity in our body against that specific infection. Okay, so that is basically the side chain theory of immunology. Cells have the receptors or the side chains, toxin will bind to that side chains, and that will give rise to more production of the side chains, which will act as a antibody and produce the immunity in our body. So from this topic, receptor ligand binding concept was also invented. Why? Because these side chains are receptors, and what is the ligand which will bind to the receptor specifically? That is the toxin. So receptor ligand binding concept was also invented from the side chain theory in immunology concept. Now the third invention of Paul Ehrlich, that is serum standardization. Serum standardization is very simple. That is, he worked with a collaboration with Emil von Behring, another eminent scientist. Okay, he actually produced and standardized antitoxin. Remember that, antitoxin, not antibody. So both of them in a collaboration, they produced and standardized antitoxin. And these type of antitoxin, like, they functions first in case of anti-diphtheria serum. So that is anti-diphtheria serum, which is basically antitoxin, and this helps in the concept of passive immune therapy and vaccination also. So passive immune therapy vaccination is actually comes from this serum standardization technique. This was the third invention. Now there is a fourth invention, that is basically in case of chemotherapy technique. Now in case of chemotherapy, there is obviously chemical compound which actually acts as a pharmacological compound. So he first one who linked or make a relation between the structure of the chemical compound with the activity, pharmacological activity of that chemical compound in chemotherapy. So this was the fourth invention. Now fifth invention is he invented improved staining technique. Now this staining technique basically helped to identify and differentiate between blood cells and bacteria. If you use same staining technique for blood cell, for bacteria, how could you understand is it a bacteria or a cell? You couldn't understand. So he first improved the staining technique of bacteria and blood cell, and also the sixth one, which is basically related with this, that is in case of modern hematology, he was able to classify with the help of this staining technique and identify different types of leukocytes. Okay, different types of leukocytes was also identified and classified by Paul Ehrlich. Okay, why? Because of this improved staining technique, he was able to identify them. And this was also the basis of Ziel-Nielsen technique to identify tuberculosis bacteria in acid-fast staining. So these are the very important contributions of this eminent scientist in immunology, medical microbiology, hematology. Thank you so much. If you like, please subscribe my channel and share with your friends on this. I'll definitely upload some shots. If you want, you can check my channel. Thank you so much.